Are you concerned about the quality of your water? In May of the year 2000, in the small town of Walkerton, Ontario, many people began to simultaneously experience stomach cramps, bloody diarrhea. In the end, over half the residents fell ill and several people even died. The culprit? Feces, a highly dangerous strain of E. coli that ended up getting into the water supply from a nearby farm. The Walkerton tragedy underscores the importance of water quality monitoring, and yet current tests are inadequate. Standard practice is to count the number of colonies of E. coli that form per 100 milliliters of water. You must wait as any E. coli in the water has been incubated and grown to detectable levels. It takes around 24 hours to get back the results of the test. How does that make you feel? By the time a positive test is returned and a boil water advisory is issued, you may have already drunk some of the fecally polluted water. Furthermore, the test returns a significant number of false results. The absence of E. coli does not assure the absence of more resistant fecal pathogens, such as many viruses. My research responds to these inadequacies. The goal? To develop a genetic test of water quality utilizing multiple markers. A genetic base test would be rapid, while the use of multiple markers would increase accuracy. To accomplish this goal, we are using a technique called metagenomics. Metagenomics is the study of a collection of genomes from a mixed community of organisms. The process we use can be seen behind me. First, we collect fecally polluted and clean water samples from watersheds across British Columbia. Next, we extract and sequence the DNA from the microbial communities within the samples. The sequences of DNA from a microbial community form what can be thought of as a genetic fingerprint. By searching for the key differences in the genetic fingerprints between the fecally polluted versus clean water samples, we can identify markers associated with fecal pollution. I've received data from the first few samples and have already managed to identify several candidate markers. I believe my research will lead to quicker, more accurate tests for water quality monitoring that will reduce or eliminate the chances of another Walkerton tragedy from ever occurring again. Thank you.
Well said.